Today, we are going to talk about the building blocks of all life, cells. According to cell theory, we know three things to be true. The cell is the basic unit of life, a cell must come from a previously existing cell, and all living organisms are made up of cells. But what makes up a cell? Are all the cells same? Well, today we are going to focus on cell membranes and two kinds of cells, animal and plant cells. Both animal and plant cells are actually pretty similar, but there are a few important differences that have a really big effect on the way they function. First, let's look at the similarities. One of the biggest between the two, and all cells actually, is that they have what is called a cell membrane. The cell membrane is a semi-permeable membrane surrounding the outside of the cell. Semi-permeable means that some things can pass through while others can't. So, what does the cell membrane actually do? Well, our initial definition said that the cell membrane was semi-permeable, and that it surrounded the cell. Its semi-permeable nature means the membrane can serve two primary functions. One, forming an outer boundary of the cell and therefore holding the cell together. And two, allowing certain substances to pass through it while prohibiting others from doing so. The first of these functions is pretty straightforward. Just like a water balloon itself holds water inside, the cell membrane holds all cellular components inside. No big surprise there. So rather than focus on obvious function, let's look at the second job, controlling entry and exit from the cell. By controlling what gets in and out of the cell, the membrane is really acting as a regulatory structure. Examples of substances that it regulates include, but are obviously not limited to, sodium, potassium, sugar, and amino acids. Each of these interact with the animal cell membrane in similar ways. Plant and animal cells have even more similarities since they are both known as eukaryotic cells, meaning they have a membrane-bound nucleus. Both plant and animal cells consist of a cell membrane surrounding many organelles, which are floating in a jelly-like substance called cytoplasm. The word organelle, which translates to little body, means each organelle has a unique job to do in the cell. Plant and animal cells contain many similar organelles, such as the nucleus, mitochondria, Golgi Apparatus, Endoplasmic Reticulum, and lysosomes. Those organelles do really similar jobs, but they have a few key differences between plant and animal cells that make a world of difference in the function of each cell. One difference is that plant cells can be bigger than animal cells. The normal range for an animal cell varies from 10 to 30 micrometers, while that of a plant cell stretches from 10 to 100 micrometers. There are three additional structures in a plant cell, and they are chloroplasts, vacuoles, and cell walls. In animal cells, the mitochondria produces most of the cell's energy for food. But plants don't eat food though, so this process is different for plants. Plant cells use sunlight for food using the process of photosynthesis. Chloroplasts are the structures unique to plants that do photosynthesis. They are large, double membrane-bound structures that contain the substance chlorophyll, which absorbs sunlight. Other membranes within the chloroplast contain the structure that actually carries out photosynthesis. Vacuoles are the storage center inside the cell. This storage unit holds nutrients, waste, and other chemicals. Vacuoles are big and can take up as much as 90% of a cell's volume. And lastly, cell walls. Plant cells also have an extra layer called the cell wall. The cell wall is outside the cell membrane and is made up of cellulose, hemicellulose, and pectin. It can be tough, flexible, and sometimes rigid. Like the cell membrane, it also provides the cell with protection, and in addition, adds structural support. Unlike the cell membrane, the cell wall does not function as a filter. So here are some differences in animal cells that set it apart. Animal cells have a lack of a rigid cell wall and allows animals to develop a greater diversity of cell types, tissues, and organs. There are some specialized cells that form nerves and muscles and tissue. This is impossible for plants to evolve. These specialized cells also give animals mobility. The ability to move about by the use of the specialized muscle tissues is a hallmark of the animal world, though a few animals, primarily sponges, do not possess differentiating tissues. Now, we've talked about some of the differences between the two types of cells and looked at the cell's membrane process. So So we're going to get a little crafty and make our own cell models.
Hi, I'm Marinda, and we are going to be making our own plant cell model. In order to do this, you are going to want some colorful construction paper, like this. You're going to want some pipe cleaners. These are going to form sort of the insides of your organelles inside the plant cell. You are going to want to have some tissue paper, hopefully to show sort of the membrane and the big center part of your cell. You are going to want a box that you will also need some green paint for. This is going to serve as your cell wall example, because plants, unlike animal cells, have hard cell walls. So we're going to put our whole thing inside of this cell wall. You can use a shoebox or just a small cardboard box. I have a shoebox. Okay, for that paint, some of the easiest stuff that you could use is probably tempera paint. And of course, you're going to want a paintbrush. I like a sponge because it's easier to get little crevices and cracks in the box. You're going to want some shiny stuff. You could use some glitter. This will help detail what's inside the organelles. You can also use beads to sort of show like the nucleolus inside of your nucleus. And for smaller bits and pieces of your cell, or for cells that have sort of a straight and long texture, I have some toothpicks that I'm going to put together to form graphite crystals. And you will also need scissors, of course, to cut out your stuff, and glue. You can use super glue or hot glue. I have some very strong stick glue that I will be using. So, your first step is going to be to cut out the shapes that you need from your colored paper. I have cut out a big green shape from my central vacuole, and I've cut out smaller shapes in different colors so you can tell that they are different organelles. So for my mitochondria, I have this. For my chloroplasts, I have this, and I'm going to use some green pipe cleaner to put stuff inside of them. And for my nucleus, I have this. So all different colors so that people looking at your cell will know what are different organelles. Now that I have my organelles cut out, I'm going to use some of my tissue paper to create the cell membrane around the edges of my hard cell wall that I have here. Here we go. So I'm going to take my glue. I'm going to glue around the edges of my cell wall in here. Then I'm going to take the strip of tissue paper I ripped up, and I'm going to crump it up into like a little skinny line. So the first side is done, and it looks kind of like this. So I'm going to keep going for all of the other edges. Kind of how it looks. See, so it's around the edges of my hard cell wall. All of my stuff is going to go inside my cell membrane, all of my organelles. Okay, so our next step is to glue down all of the outlines of our organelles, remembering that they are color-coded. So my bigger pink one is going to be my nucleus. Then I have a few of these guys. They're going to be in my chloroplasts. And over here I have my mitochondria. I have a few of those. So we're going to glue those ones down now. Okay, so now I have some of my major outlines inside my box. Okay, I'm going to use my pipe cleaners to add some detail inside of my organelles. So here we go. Alright, so I've got some of my pipe cleaners to show detail inside my organelles. You see those squiggly wigglies. Uh-oh, one of them came off, but I'll glue it back down. Next, I'm going to build my Nucleolus inside of my big pink nucleus. I have some orange pony beads that I'm going to be using for my Nucleolus. So, I'm going to glue these down. Okay, so now I have my Nucleolus inside of my pink nucleus. Now I'm going to put in my Golgi Apparatus. Again, this, if you have room, you're going to try to fit it outside of your vacuole. Okay, so you can see in the orange pipe cleaner over there, I made my Golgi Apparatus. Alrighty, we're going to make my smooth endoplasmic reticulum. I'm going to use my blue pipe cleaner for this. Now, because it's smooth, I'm not going to have any ribosomes inside of it. Just this. Okay, so I have my smooth endoplasmic reticulum on one side of my nucleus, and now I'm going to add my rough endoplasmic reticulum. Okay, with my rough endoplasmic reticulum, just to represent some ribosomes, I'm going to use some more pony beads, this time yellow ones, and I'm going to glue them to that side so it shows what it is. So, if you can see in my nucleus, on one side a smooth endoplasmic reticulum, and on one side the rough one with a couple of ribosomes in there to show you what it is. Okay, on my next step, I am going to add some graphite crystals to the inside of my vacuole. As I said earlier, I'm going to use some toothpicks to represent those. Okay, I have my graphite crystals in here. And my next thing is the Druce crystal. I'm thinking I'm going to use some white tissue paper and kind of crumple it up and set it in there in a ball shape to represent my Druce crystal. Okay, my Druce crystal is in there as that white tissue paper. Okay, depending on how detailed you want to get, you can also throw in some glitter to sort of represent your cytoplasm around the outsides of your vacuole. So, here is my plant cell example. It might be hard to see, but I glued some glitter around the edges of the vacuole to kind of show the cytoplasm. Your next step could be to use a pen or a marker to go through and write a label beside each of your organelles, or you could find little labels on sticks and stick it in there showing what each item is. Always remember, cell walls are special for plants. They're made up of cellulose. Chloroplasts are special because they are where photosynthesis happens. And the large central vacuole is special to plant cells as well. Okay, I hope you had fun making this. Again, you can label it either with a marker inside the cell yourself, or you can find little labels that you can stick into everything. Have fun.
All right, for our animal cell model, the only things you're going to need is going to be a bunch of construction paper of different colors, pencils, scissors, some cardboard, a hot glue gun or glue stick, and maybe some markers. All right, let's get started. Okay, so we're going to go ahead and make our shape of our cell. We're actually going to be doing a cross section. So I'm using a plate to create this circle with, so I'm just going to go around and trace my plate and then cut that out of the cardboard. So you can either go ahead and cut that down the middle, that's what I did, or you can go ahead and cut your circle out and then cut it down the middle. But by the end of it, we want 2 halves of a circle. So next I'm going to be creating my cytoplasm. And so I went with a light blue for my cytoplasm, and that is going to be the jelly that holds all of our organelles together. So you're going to want to trace your 2 halves of your circle with your color of cytoplasm paper. Then we're going to move on to our cell membrane. So we know that animal cells do not have a cell wall, but they do have a cell membrane. So I am picking a darker shade of blue and just tracing about a 1/2 inch around my cytoplasm. So this will represent that it is enclosed, but it is not a rigid cell wall. So once you have that traced out, you're going to go ahead and cut it and also glue it to your cytoplasm, whatever color that may be. All right, so now it is time to go ahead and attach your cytoplasm and your cell membrane to your cardboard. So I am just taking some hot glue and really, putting a lot on there and placing my construction paper onto the cardboard. Now, if you're using hot glue, make sure to be careful. We don't want any burns while doing our DIY. So now it's time to turn our cell membrane model into a cross section. So what you're going to do is take those 2 half circles and put them perpendicular to each other. So I'm just taking a little bit of hot glue and placing it on the base of one of my half circles, and then I will hold it to the base of the other half and let it dry for a little bit. And once it has dried, then we are going to kind of have a cross section of our animal cell. And then we will move on to making our organelles. All right, our first organelle is going to be the nucleus and the nucleolus. So I'm just taking a circular object and tracing around it for the nucleus, and that's going to be one color of construction paper, and then I'm going to grab another color for my nucleolus, which is going to be smaller but placed in the center. So I'm going to cut those out and glue them together and then cut them in half. So while we do that, let's talk about the science. So the nucleus is a highly specialized organelle that serves as the information processing administrative center of a cell. This organelle has 2 major functions. It stores the cell's hereditary material, or DNA, and it coordinates the cell's activities, which include growth, metabolism, protein synthesis, and reproduction, or cell division. Now, the nucleolus is a membraneless organelle with the nucleus that manufactures ribosomes, which ribosomes are the cell's protein-producing structures. So through a microscope, the nucleolus looks like a large dark spot within the nucleus. A nucleus may contain up to 4 nucleoli. So now that we know the science, it's time to go ahead and attach our nucleus to the model. So you're going to cut down the middle of that smaller circle and do exactly like we did before, and take your hot glue or glue stick and glue one to the base and one perpendicular to the other part. So our next organelle is going to be the endoplasmic reticulum, and there's actually 2 different kinds of these. So you're going to draw out your endoplasmic reticulum, and you're going to do 2 of those in whatever color you want. So then you're also going to just cut those out, and with one of them, you're going to add an extra special texture to it, but we'll get to that in just a second after we talk about the science. So the endoplasmic reticulum is a network of sacs that manufacture, process, and transport chemical compounds for use inside and outside of the cell. It is connected to the double-layered nuclear envelope, providing a pipeline between the nucleus and the cytoplasm. And there are 2 basic kinds of endoplasmic reticulum, rough and smooth. The surface of the rough endoplasmic reticulum is covered with ribosomes, giving it a bumpy appearance when viewed through the microscope. This type of endoplasmic reticulum is involved mainly with the production and processing of proteins that will be exported and secreted throughout the cell. So now it's time to differentiate between our smooth and rough endoplasmic reticulums. So what I'm doing is taking a different color and using a hole puncher and just punching out a bunch of holes. So these will actually go in two different parts. Some of them will go onto one of our endoplasmic reticulums, and the others are just going to be placed around our cytoplasm as ribosomes. So let's talk a little bit about ribosomes. So ribosomes are in all living cells, and they are tiny organelles composed of approximately 60% RNA and 40% protein. In eukaryotes, ribosomes are made of 4 strands of RNA. In prokaryotes, they consist of 3 strands of RNA. Pretty interesting. Also, don't forget to make an example of the smooth endoplasmic reticulum. Our next organelle is going to be a vacuole, which, as we learned earlier, all cells have vacuoles in them. Up next are lysosomes. So the main function of these microbodies is digestion. Lysosomes break down cellular waste products and debris from outside the cell into simple compounds, which are transferred into the cytoplasm as new cell-building materials. Now next is our mitochondria. Now I'm not going to lie, this one definitely took me the longest to create out of paper, but with a little patience, you can do it. So let's talk the science. Mitochondria are oblong-shaped organelles that are found in the cytoplasm of every eukaryotic cell. In the animal cell, they are the main power generators, converting oxygen and nutrients into energy. So now we have our Golgi apparatus and our Golgi bodies. So the Golgi apparatus is the distribution and shipping department for the cell's chemical products. It modifies proteins and fats built into the endoplasmic reticulum and prepares them for export to the outside of the cell. And last but not least, we have our centrioles. So centrioles are self-replicating organelles made up of 9 bundles of microtubes and are found only in animal cells. They appear to help in organizing cell division, but aren't necessarily essential to the process. I think that they look cool, so that's why I added them. All right, so now we have gone over all of the science and made all of our organelles. It is time to attach them to our model. So all you're going to do is just take your glue or hot glue gun and start attaching. Once again, if you're using hot glue, make sure to be careful. I hope you have enjoyed our video today, and if you make either one of these DIY models, please share your pictures with us on Facebook or Instagram. Thank you. Bye.